about ARCHS4, and then I do some slides, and then I go to the website and show it live. So ARCHS4 is a large data processing project that mainly really tries to tackle the cost of aligning RNA-seq data to human and mouse reference genomes. And the main issue is that there is a lot of factors that really drive the costs of such analysis. So one is that it's fairly CPU intensive. So we want to use cloud resources, and their trade-off is that they are, very elastic, so you can just get them when you need them. But while you utilize them, they're way more expensive than a system that you would purchase. So pretty much if you would rent a machine at Amazon for a year, you could buy the whole system outright. The same goes for memory. Memory on these machines is very expensive. But then the big advantages really from the cloud are that you have this amazing storage, which is fairly affordable and elastic, and you have very significant network bandwidth. And this is very important because we're going to touch on how much data we actually have to process for this. So just real brief, the architecture of ARCHS4 is, as I mentioned before, cloud-based. So we have a dedicated control server that hosts an API to which the clients of the ARCHS4 workers can come and receive job descriptions. And they basically run a Dockerized container with a predefined workflow. And so mainly for the ARCHS4 workflow, it is to retrieve the FASTQ files, align them to a specific reference genome, and then upload that data into a cloud repository. So over time, the data really started to accumulate. So we started this project in 2017. So at that time, there were maybe 100,000 samples of RNA-seq for human and mouse. And by then, it is significantly increased, as you can see on the top right. And so there's always the time delay with the samples when they become available. So it's not really a drop-off in samples. So this will just start going up, right? So the cost is a really important aspect. And so far, we aligned nearly 20 trillion reads, which is significantly more than a petabyte of data. So you can imagine it's about like 1 million gigabytes. So we're like kind of getting in the territory of what like Netflix, let's say, does, right? When they stream, like 1 million movies at the same time. This is kind of the scale of this project. And so it's more than 1 million samples that we aligned from 30,000 experiments. So this is just like a quick breakdown. We have about 600,000 mouse samples and 700,000 human samples. And then we also have a couple of samples from other species, but that's like a much smaller data set from like 6 of the most common RNA-seq data sets for other species. So the resource itself basically just tries to build large pre-aligned gene count tables that people can use. So we use this H5 data format, which is nice for programmatic access. But then we also have APIs that try to serve the data on request. And so we pull metadata from GEO and then also inject that into those data files. So this data, like we really started to do this project because we wanted this data for our own purposes. So you can do a lot of stuff with that. So you can build gene expression atlases and tissue and like cell lines, cell line expression, and you just get that from this heterogeneous source of data, which I think is a big advantage of resources like GTEx because they're very homogeneous. So that's on the one end maybe an advantage, but on the other end you lose a lot of the biological complexity that you encounter in real data and like the differences you will observe from different labs doing similar experiments. So we also use this data to do gene function prediction, as I mentioned before in the gene shot presentation. And so it's a pretty wealthy like resource that we use for like many of our projects. So you can do fun things like also look at how many reads, the fraction of reads that actually align in a FASTQ file given different institutions that publish the data. So there's some interesting stuff to find. So the ARCHS4 data set is really supposed to be a resource that is to be used by other tools, right? And it's just making the ease of access to this data much easier, right? Because let's say you have this idea, oh, there's this experiment, they did this RNA-seq analysis. You really want really quick access to it. You don't want to start aligning it and then realize you can't use the data. So, this should make it much easier to reuse existing RNA-seq data. So there's tons of interesting tools that use this data already. So not going into too much further. And then so for future plans, so so right now we did only cumulative updates to the existing aligned reads. So we'll probably do a complete reanalysis with the new ensemble version that will come out hopefully this year. So we're still waiting for that to happen. We are actively working on reducing the cost even further. So we're about at ½ cent per sample right now. And we also want to focus more on single cell. So there's a lot of single cell data. So that's coming up. And then we also want to build some improvements in building companion packages for Python and R, so you can integrate that easily. So let me go to the website. So this is the landing page of ARCHS4. So it's just showing the workflow I showed you before. We have the statistics. This real quick, this is the ARCHS4 zoo, which contains the other species. So this is just supporting downloads of the bulk files. And we'll probably update this in the future. But so getting started, so this is the main page for the data view. So here we have human data selected, and we're looking at the samples. So this is a 3-dimensional representation of all the samples. So each point is an individual sample. And this is a t-SNE projection of that data. So you can see that the data is quite structured, and that's, I guess, what we want to see. So now we can actually search the data in real time. So we can look for macrophages, for example, in the human data. So we can see that there's a lot of macrophages in this area here. There's a cluster over here. So yeah, so we can do this, and then down here in the selector, we get this view. So we found 3,500 macrophage samples, and we do this by searching the GEO metadata. And this is from 150 different experiments. So it's pretty encouraging that they all seem to cluster together. We can also search for series. So here I looked for a series, and this is already pre-annotated in BioJupyS. So if I go in here, I can directly import the data to BioJupyS, which is another tool developed in the Mayan lab. And it allows us to potentially do differential gene expression analysis and more complicated downstream stuff. Yeah, so then another brief thing to do is, so here I can also search for signatures. So let's say if I go to the mouse data and I try to search signatures that match these up and down regulated genes. This is what I get. So I find this cluster here. So these samples seem to match this input query, but there's like some scattering all around. And this can happen because we're not looking for like full signatures with all of these sets. So it's like always a little bit fussy if you search like this. We also have enrichment, where you can look for potentially upregulated transcription factors. So let's see if this returns something. So here we found about 270 samples that match a transcription factor from this GEO library, and we have like different libraries in here. I guess arguably the most important side is the download section. So this is the data that we offer for download. So this is the newest version that just came out like 2 weeks ago. And they get pretty large, so they're about 17 gigabytes. But it's heavily compressed data, and you don't have to unzip the whole file. You can just programmatically get to exactly the data that you want. And so we support gene level counts, transcript counts, and TPM data. TPM data is already 100 gigs, so beware. We also have expression data from Affymetrix arrays. So there will be in a different size because the probes don't cover all the genes that we detect with RNA-seq. But we added them in the same format. So if you have a script that accesses these H5 files, you can also just access that stuff. And then there's some other stuff that we have. And then we have also, for this, we have an extensive help section. So we have a YouTube video that goes through the website if you want like a more in-depth introduction to it. And so this also shows all the metadata. So this is kind of the inside of an H5 file. Looks a bit scary, but in the end, this is the important field that you're interested in. So this is the data expression. And you can kind of see the number of samples that we have and the number of genes. And then in the metadata, you will find, you know, all the sample information, the accession IDs, stuff like that. Yeah, I think that's more or less it that I wanted to show. Is there like something small? Oh yeah, right. So we also have gene pages. So if we search for an individual gene, let's say SOX2, we get like an overview of predicted gene functions. I guess this is also very similar to what we now have in GeneShot, and that's probably more comprehensive than this. But this also has the tissue expression atlas and the cell line expression atlas. So if you just want to see in what tissues the gene might be expressed in, at what variation, like variants, this could be an interesting resource. And yeah, so that's it for me. And then